4, which is to describe the process of neural integration and the role of the axon hillock in this process. So the drawing that I was trying to illustrate actually gets into neural integration. So we can discuss this here, but definitely reference it after, as I'll share it with the recordings today. So let's think about neural integration and what that means. Now, there are a few different processes involved in neural integration. Summation, we've talked about the two types of summation, the fact that we can sum graded potentials if they're close enough in time and if they're at the same synapse via temporal and spatial summation. So that's one aspect of neural integration. Frequency coding, which is what we were describing earlier, this is where multiple action potentials is a way to send a stronger signal. So if the signal being sent is graded as a strong signal, that means that it is going to result in more frequently produced action potentials, which is going to result in more neurotransmitter being released, which is going to result in more of that neurotransmitter binding to receptors and bringing about a stronger signal on the postsynaptic side. So frequency coding is the way that we appreciate different types of action potential signals. Divergence and convergence, right? This is just looking at the ways in which signals can be diverted, right? So you can spread one synapse, the signal there across several different axons, and vice versa. Convergence, which is where several axons can result in a single synaptic signal. And we'll illustrate what those two means, but those are also involved in neural integration. And then the axon hillock is where all of this information is assessed. And if it is enough of a signal to get to threshold, which is -55, then we start the action potential at the axon hillock as well. Okay, so neural integration is basically adding all of the signals that are arriving on the soma, adding that up at the axon hillock and determining whether or not it is enough of a membrane potential change that gets us to threshold. And if it is, automatically we fire an action potential. And if it isn't, then there is no action potential that will be initiated. So the neural integration is the process of determining if the membrane potential change is strong enough to begin an action potential. Okay. Divergence and convergence. So looking at the ways in which the signal can either dissipate and spread out. So here's one presynaptic neuron, here's its axon, and then notice that it has several axon endings that are synapsing onto different neurons. So here the signal is sort of spreading out or diverging onto different neurons via different synapses. The opposite is convergent. So we can also have many axon terminals synapsing onto a single soma, okay? And that's a way just to strengthen the signals that are coming in on this one postsynaptic neuron. So that's to say that there are billions of synapses happening across the nervous system, okay? We have neurons signaling other neurons, one neuron signaling many neurons, but also each individual neuron receiving multiple synaptic connections. And then we also have neurons signaling other types of cells as well, muscle cells, gland cells. And so there are billions of synapses happening within the central and peripheral nervous system. Summation. Now, summation we talked about earlier as well. This is another type of analysis that's happening at the axon hillock to determine if we have signals that are strong enough to get us to threshold. So we can add the IPSPs and EPSPs. Remember, IPSPs are negative membrane potential changes. EPSPs are positive membrane potential changes. Another way to say that IPSPs are hyperpolarizing, EPSPs are depolarizing. So if we think about the types of summation, temporal summation, for example, this is if we have one synapse that's happening close enough in time. So if we take in this neuron, here are 3 different presynaptic neurons, and then here's the postsynaptic neuron. If we're only taking into account synapse A, and we have 2 synaptic signals arriving at synapse A that are close together in time, we can add those together, and it could be a strong enough signal when we add them to arrive at -55, and then we can fire an action potential. So this is temporal summation. One single synapse being considered, and the signals coming in are so rapid that they're close together in time, and we can sum them for a stronger total signal. Spatial summation is similar, but now we're looking at several different synapses. So in the example down here, we're looking at presynaptic neuron A, B, and C, all communicating with postsynaptic neuron. And so we can add these signals together because they're coming in at the exact same time. Different synapses, same exact time. And so we can sum those together to result in a stronger membrane potential change. So the example here, we look at synapse A, which is depolarizing. B is also depolarizing. C is hyperpolarizing. So C is actually an IPSP. However, A and B alone are enough to generate an action potential. If we add C, C actually cancels out A or B, and so that is not enough to bring us to a threshold. Okay, so we can sum these various synapses if they are happening at the exact same time in different spots, and that is spatial summation. Okay, and then the third mechanism here for neural integration, I'm sorry, someone's asking which one is C, so apparently my mouse wasn't showing that properly. Okay, so for spatial summation, we said synapse A was a depolarizing event, synapse B also depolarizing, synapse C, notice it's a negative membrane potential change. So considering A and B together, that gets us to threshold. But when we consider A, B, and C, that cancels out, right? Because C is negative, cancels out B, and then we end up only having A, which is not enough for a threshold, right? Not enough for an action potential. All right. Okay. So the final component here, frequency coding, this is where we think about the summing, and then we also want to think about the frequency of action potentials. If we have many action potentials arriving at the axon hillock, then we have a stronger signal being generated because we now have a faster rate, which means that more neurotransmitter is going to be released at the axon terminal, and that's going to result in a stronger signal on the postsynaptic side. Okay, so this is another way to say that more action potentials means more neurotransmitter release, means a stronger graded potential on the subsequent neuron. Okay. We also want to think about presynaptic modulation. And this is using an example of axoaxonic synapses. If you remember, we talked about various types of synapses. We had axodendritic, which is the most common. We had axosomatic. And axoaxonic is when one axon terminal is synapsing onto another axon terminal. So this can be a way to modulate the synapse that's happening between presynaptic neuron and a postsynaptic neuron, right? We have a third modulating neuron that is going to either encourage, which is facilitation, or inhibit, right, inhibition of the presynaptic neuron in terms of the amount of neurotransmitter that is released. So this can be a way to further strengthen the signal or to inhibit the signal of this presynaptic neuron by the third modulating neuron that is synapsing at this terminal. Okay, so let's describe what that looks like. So when we think about presynaptic facilitation, so here we have neuron C synapsing onto neuron X. And then neuron E here is the modulating neuron. Now, because this is facilitation, right, presynaptic facilitation, Neuron E is going to encourage neuron C to release more neurotransmitter at that synaptic cleft. And so what that does is it generates a stronger signal on neuron X, right? Which means a stronger graded potential on neuron X, which is going to be close enough or meet that negative 55 threshold and actually allow an action potential to be initiated. So this is a way that a third modulating neuron can encourage the synaptic neurons to release more neurotransmitter and result in a stronger signal. However, I want to make it clear that the modulating neuron does not, on its own, fire any action potentials. The modulating neuron has no membrane potential change. So if we look at neuron C on its own, there is a small depolarizing change. Neuron D on its own, also a small depolarizing change. Neuron E on its own has zero membrane potential change because this is not resulting in any electrical impulse or a chemical impulse. All that it's doing is it's telling neuron C to release more of its neurotransmitter, right? But it has no other membrane potential change. However, neuron C plus E, because of that facilitation, now we have a stronger greater potential, which is reaching negative 55, and then we can have an action potential. Neuron D and E will be right, no action potential because neuron E is not modulating neuron D. Okay, so if we considered both of those neurons, it would be just as if D were synapsing alone, right? Because D is not being facilitated by modulating neuron E. Okay, are we good so far? Any questions on facilitation? So two things to bear in mind. Neuron E is the third modulating neuron, has no membrane potential change on its own. However, because it is modulating neuron C, it does allow neuron C to result in more neurotransmitter release, i.e. stronger graded potential, which allows us to get to an action potential. Okay, let's look at presynaptic inhibition. And this is the opposite, but the principles are the same. We're looking at a third modulating neuron that is synapsing at neuron F in this instance, the synapse between neuron F and neuron Y. And the inhibiting neuron here is actually going to block or decrease the amount of neurotransmitter coming from neuron F. Okay, so we can see that when neuron H is engaged, the amount of neurotransmitter being released here has decreased, which tells us that there's going to be a smaller signal or a smaller greater potential being released, and that is going to inhibit or dampen the synaptic response at neuron F and Y. So let's look at these neurons on their own. If we look at neuron F, a depolarizing event, neuron G is also depolarizing. Neuron H is our modulating neuron, so it has no membrane potential change on its own. And then if we consider neuron F and H, notice that it sort of cancels out the depolarizing event that is otherwise seen at neuron F, okay, at synapse F. So F plus H dampens that response, and we have no change in that membrane potential. If we consider neuron F plus G, they're both depolarizing. And because we're not considering H, they are both going to be a stronger impulse that actually results in action potential. But if we do consider all three, F, G, and H is going to cancel out F, right? Because it's going to dampen the response of F, and then it's going to be the result of just G alone, right? Only a small depolarizing event. Okay. So that is presynaptic facilitation versus presynaptic inhibition, and the way that that modulating neuron can either inhibit or facilitate the synapse at the neuron that it's modulating. Okay. So just to describe this further, the type of synapse is axo-axonic. It can either excite or inhibit another synapse that's already happening. And it is selective, right? This is what we mean by only one modulating neuron can basically modulate an existing synapse. So the modulating neuron is selective to the single synapse that it is modulating, whether it's inhibition or facilitation. The other types of um, synapses we describe, axodendritic and axosomatic. These are excitatory or inhibitory to the postsynaptic neuron. So these are going to result in membrane potential changes. Remember, we said that axodendritic is the most common type of synapse, and this is the axon terminal synapsing onto the dendrites or the soma, respectively, of the adjacent neuron. And this is non-selective. That means that as long as the axon terminal in question is coming into contact with the dendrite or soma in question, there's always going to be the transmission of that chemical synapse, right? It's not selective for a specific synapse. The presynaptic facilitation or inhibition is selective for a specific synapse. Now, just to sort of wrap up and summarize what we've described in terms of neurotransmitter release and the synapse, right? The events happening at the synapse, we want to kind of zoom out and think about how all of the neural integration factors are going to be taken into account, and we're going to assess what's happening with our neuron at the axon hillock. So we think about our presynaptic neuron. And these are the axon terminals of the presynaptic neurons. And notice that we have a few that are synapsing at different spots on the soma here. Okay, all of this results in neural integration. We're going to take into account the type of synapse. We're going to take into account the amount of neurotransmitter that's being released and bound to receptors. We're going to take into account the frequency coding, right? How fast are those action potentials arriving, resulting in more neurotransmitter. And we're going to take into account the type of channel that is opening up or closing on our postsynaptic neuron. So all of those things will be assessed, and then at the axon hillock, that will determine whether we have enough of a signal to start an action potential, and then it's going to propagate down the axon, and the very same thing is going to happen all over again, right? The action potential arrives at the axon terminal, and then we determine the strength of that signal to cross the synaptic cleft. Okay, so we want to kind of zoom out and remind ourselves that while we've been looking at one neuron to the other, there are billions of synapses happening, going back in different directions. There's convergence, there's divergence, there's frequency coding. There's a lot of different information that is happening that is determining the types of signals and the direction of signals. Okay. I also wanted to spend some time here recapping the location of different receptors and how they bring about different responses. So we talked about ligand-gated receptors. Those are typically found on the soma of the postsynaptic neuron. And ligand-gated means that something has to bind, which makes sense because a neurotransmitter is being released from the presynaptic axon terminal, and it's going to have to bind to some ligand-gated channel in order to bring about a cellular response. We talked about sodium, voltage-gated sodium channels and potassium channels. Those are concentrated along the axon. So those are going to be more so found along the axon, and they're going to help in the propagation of the action potential. And then we talked about calcium channels, voltage-gated calcium channels that are found at the axon terminal. So both here and here, and they are responsible for the release of neurotransmitter. So those voltage-gated calcium channels are going to be more concentrated at the axon terminal itself.